Hi guys, kamusta? We are Group 3 and we're here to present Penicillium fungi. So take the floor, Tere.
Thank you, Francisco, and I'm Teresa Halatot presenting our introduction. For a brief background on Penicillium, it is a genus of Ascomycetes fungi, commonly referred to as blue or green mold, where it has about 350 species. The genus is considered as a saprophytic fungus where it obtains nutrition from dead or decaying materials. Species within the genus can be observed in different environments such as in soil, air, decaying fruits, and others. They are well known for the extraction of penicillin, which is one of the most important antibiotics discovered by Alexander Fleming. Moving on to the basic morphology of penicillium, we now look into its morphology under the microscope. It has septate hyphae, wherein the hyphae of this genus are divided by the septa into cellular compartments. It has simple or branched conidiophores with phyalides attached at the end, forming brush-like clusters where unicellular round-shaped conidia can be found in chains. Penicillium in culture media can be described with colonies large in size, has a filamentous and irregular form with colors that range from white, gray, and green. Colonies with a white margin and pigmentations like yellow and reddish spots on the colony can be seen as well. The elevation of the penicillium colonies can be ungunate and some may be raised. They are also characterized with the powdery or velvety texture. Now for the isolation techniques, we now have Mr. Haranilia presenting the flow chart on the protocol of isolating penicillium.
Thank you, Teresa, for that introduction. Good day, I am Jeffrey Sierranilla, and I will be discussing the flowchart that will be followed for the isolation of Penicillium species. The flowchart involves four major steps: the preparation of the soil sample, preparation of ciliary dilution, incubation, and the isolation of Penicillium species. For the preparation of the soil sample, the initial moisture content is computed first. Water will then be added to increase its moisture content up to 10%. The soil will then be transferred into a container and will be covered with a plastic wrap that has tiny holes to allow aeration. The container containing the soil will then be weighed and will be given 1 week for incubation to allow the fungi to grow. After 1 week, the weight loss caused by dehydration will be calculated. Water will then be added to bring the weight back to its original number. The soil will be mixed with distilled water in preparation for 110 cereal dilutions. 0.1 ml from the dilutions will be taken and be spread over the sterile rose bengal streptomycin agar. The agar plates will be given 1 week to incubate. After 1 week, the agar plates will be scanned for the presence of Penicillium species and their colonies will be counted. The identification of Penicillium species will be done using the manuals of Rapper and Tong and Gilman. The identified colonies of Penicillium species will then be subcultured to prepare pure culture of the Penicillium species.
Thank you, Troy. Now, I am Jasper Jimenez, and we will now proceed to the culture methods utilized for the Penicillium species. First, we have the direct plate method. The direct plate method is easily the preferred method for detecting, enumerating, and isolating fungi such as Penicillium. This involves placing food particles or items of interest directly onto the solidified agar media, while on the other hand, we have the dilution plating method. It is more labor intensive as the sample needs to be diluted several times. On the next slide, we have the different culture media for the Penicillium species. First, we have the dichloran Rose Bengal chloramphenicol agar. This agar is composed of rose Bengal, dichloran, chloramphenicol, peptone, potassium dihydrogen phosphate, epsomite, agar, and distilled water. This agar has a pH ranging from 5.5 to 5.8. Next, we have malt extract agar composed of malt extract, peptone, glucose, agar, and distilled water with a pH of 5.6. Chapek yeast extract agar, on the other hand, is composed of potassium dihydrogen phosphate, Chapek concentrate, trace metal solution, yeast extract, sucrose, agar, and distilled water with a pH level of 6.7. Lastly, Penicillium species is also cultured in media such as Chapek dox agar, which is composed of sodium nitrate, potassium chloride, epsomite, iron 2 sulfate, heptahydrate, and distilled water. And the commonly known potato dextrose agar that consists of potato extract, dextrose, agar, and distilled water.
Thank you, Jazz, for that wonderful presentation. And now let's proceed with the guide questions. So for the guide question number 1, we were asked for the environmental use, economical use, and potential application of penicillium. So for the environmental use, penicillium was shown to exhibit a high proportion of endoclocanase, beta-glucosidase, and other protease activities. These enzymes are known as decomposers for protein and cellulose materials in the intertidal zone. So in short, penicillium are ecological decomposers. Another environmental use of penicillium is through the degradation of pollutants. They can degrade environmental pollutants like synthetic dyes and polycyclic aromatic hydrocarbons or PAHs. So for the economical use of penicillium, it is heavily used in the pharmaceutical industry. They are used to produce antifungal drugs, immunosuppressants, and antibiotics. Another economical use of penicillium under the food industry is through the fermentation of cheese and production of meat products like sausages. So for the potential application of penicillium, Penicillium sp has the ability to produce antibacterial properties called penicillin. So with this in mind, we can hypothetically say that Penicillium sp has the potential to be integrated in the field of agriculture as a bactericide. So for the second guide question, we were asked for the specialized reagents and laboratory techniques used for penicillium. So for the specialized reagents, we use the rose Bengal dye. And for the laboratory technique, we use the conventional agar plate method using rose Bengal agar, Chapek yeast extract agar, and malt extract agar. And for the specialized laboratory technique, we're able to search that utilizes the use of motor chamber that ferments the penicillium, wherein it is being sanitized, maintained with proper conditions, and supplied with essential nutrients. This is used for the production of penicillin in the antibiotic drug. This can be further be illustrated in the image above me. So to answer the guide question, a rose Bengal dye is necessary to penicillium growth because of its properties of inhibiting bacterial growth while maintaining a decent amount of fungal colony for study. So it is really suitable for fungal species like Penicillium sp. So that's all for the guide questions. So here are the reference used for our contents and images. Once again, we are Group 3 for Penicillium fungi. I am Isidro Francisco III, and together with me is Ms. Halatot Teresa, Mr. Haranilia Troy, and Mr. Jimenez Jasper John. That's all. Salamat and ingat lagi. Watermelon sugar high, watermelon sugar high, watermelon sugar high, watermelon sugar high.